The overall goal of this procedure is to investigate various stages of the hepatitis C virus replication cycle. This is accomplished by first generating HCV genomic RNA transcripts from linearized HCV plasmid constructs. The second step is to transfect the cells with HCV RNA. Next, the cells are plated for various time points and assays. The final step is to collect the cell culture supernatant for measuring viral titer and harvest transfected cells for protein and RNA. Ultimately, reverse transcription PCR, Western blotting, immunofluorescence assay, and HCV titer are performed and demonstrate a robust HCV infectious cell culture system.
The main advantage of this infectious hepatitis C virus cell culture system over the HCV replicon system is that viral entry, virion assembly, and egress steps can be investigated. This protocol can help answer the key questions in the hepatitis C virology field, such as virion morphogenesis and host-pathogen interaction. The implications of this technique extend toward vaccine development, as it allows characterization of attenuated viral strains that can be evaluated as potential vaccine candidates.
Though this method can provide insight into hepatitis C virus, it can also be applied to other RNA viruses in the family of Flaviviridae. Generally, individuals new to this method will be faced with the challenge of generating high-quality HCV genomic RNA. Studying the complete replication cycle of HCV became feasible in cell culture following the discovery of a genotype 2A HCV Japanese fulminant hepatitis 1 isolate.
Visual demonstration of virological assay is critical because the assays require expertise in both molecular and cellular techniques.
Using an intragenotype 2A chimeric virus, FNX-HCV, and FNX-HCV pol null HCV for evaluation of viral replication, linearize previously generated viral plasmids by digestion with XBA1 restriction enzyme in a 2 ml tube. Then, treat the plasmids with mung bean nuclease to generate blunt ends. Purify the digested plasmids by anion exchange chromatography. Verify the integrity of the linearized plasmid by subjecting DNA to agarose gel electrophoresis. Next, add the linearized plasmid DNA template into a 0.2 ml tube containing T7 RNA polymerase reaction components to synthesize HCV genomic RNA transcripts. Purify the newly synthesized DNase-treated RNA using an RNA purification kit. Then, verify the RNA production by agarose gel electrophoresis. Following this, quantify the RNA by spectrophotometry. Detach Hu7-based adherent cells using trypsin enzyme treatment and collect the cells into a 50 ml conical tube. Following centrifugation, resuspend the cell pellet with cold, low serum media. After repeating the centrifugation, resuspend the cells in low serum media at 1 × 10⁷ cells per milliliter. Next, mix a total of 10 micrograms of transcribed viral RNA with 400 microliters of resuspended cells into a pre-chilled 0.4 cm electroporation cuvette. Deliver the viral RNA into the cells using an electroporator at 270 volts, 100 ohms, and 950 microfarads. When finished, resuspend the electroporated cells in 10 milliliters of complete growth media with 15% FBS. At this point, plate the cells in both T25 flasks at about 1.2 × 10⁶ cells per flask and 48 well plates at 1 × 10⁴ cells per well for 4, 48, and 96-hour time points. Replace the media at 4 to 8 hours post-transfection with fresh supplemented growth media with 10% FBS to remove dead cell debris from the cultured flasks and plates. Using a serological pipette harvest the cell culture supernatants at the 48 and 96-hour time points into a 15 milliliter conical tube. Then, remove cellular debris from the collected samples by centrifugation at 15,000 rpm for 10 minutes at 4 degrees Celsius. Following centrifugation, store the cell-free supernatants at -80 degrees Celsius. Lyse the cells for protein and RNA analysis by Western blot and reverse transcription quantitative PCR or RT-qPCR at the indicated time points. Reverse transcribe 1 microgram of total cellular RNA using reverse transcriptase enzyme and a specific primer for the HCV sense strand that binds to the 5' untranslated region in a 0.2 milliliter tube. Also reverse transcribe FNX HCV RNA of known genome copies using an HCV sense strand primer. Using a real-time PCR system, carry out qPCR by using 50 nanograms of the resulting transcribed cDNA, using specific HCV primers and DNA-binding green dye containing qPCR supermix. Use the following conditions when running qPCR to determine the HCV RNA copy number. Following qPCR, resolve the cell lysate from the viral RNA transfected at 96 hours post-transfection using SDS page. Then, transfer the resolved proteins in the gel to a polyvinylidene difluoride membrane by transblot turbo method. Block the membrane using a blocking solution containing 5% skim milk and 0.2% tween 20 in PBS and placed in a container. Incubate the membrane with primary mouse monoclonal antibody NS3 at a 1 in 1000 dilution and beta-actin at a 1 in 5000 dilution in a 4 degrees Celsius cold room. After incubation, add goat anti-mouse immunoglobulin G conjugated to horseradish peroxidase at a 1 in 5000 dilution and detect by chemiluminescence. At this point, fix the HCV RNA transfected cells using methanol for 30 minutes at -20 degrees Celsius for the immunofluorescence assay. When finished, wash the cells with PBS 3 times. After blocking with immunofluorescence assay blocking buffer, use rabbit polyclonal anti-NS5A primary antibody and mouse monoclonal anti-dsRNA antibody J2 at a dilution of 1 to 200 and incubate for 5 hours to overnight in a 4 degrees Celsius cold room. Following incubation, wash the cells with PBS 3 times after the primary antibody. Then, add goat anti-rabbit immunoglobulin G 488 polyclonal secondary antibody and goat anti-mouse immunoglobulin 594 polyclonal secondary antibody at a 1 in 1000 dilution and incubate for 1 hour at room temperature on a tabletop rocker. After washing the cells with PBS 3 times, stain the nuclei using Herx dye and view using a fluorescent microscope. Next, plate naive Hughes 7.5.1 cells at approximately 3 × 10 to the 3rd cells per well using a 96 well plate. The next day, perform tenfold serial dilutions of cell-free culture supernatant harvested from HCV RNA transfected cells using growth media and inoculate in triplicate onto Hughes 7.5.1 cells. Fix the cells at 72 hours post-infection using methanol for 30 minutes at -20 degrees Celsius. After removing the cells from the freezer, immunostain for HCV NS5A protein using the previously described conditions. Using a fluorescent microscope, count the NS5A positive cell foci in the well with the highest viral dilution and calculate the average number of focus-forming units per milliliter. The linearized HCV plasmid quality was assessed by gel electrophoresis. The HCV DNA was subjected to T7 RNA polymerase-mediated in vitro transcription, which yielded a single RNA product at 9.6 kilobases. RT-qPCR results indicated that the wild-type virus replicated the genome efficiently. The wild-type exhibited a 1 to 3 log higher level of genome replication compared to pol-null virus. The wild-type virus produced the NS3 protein involved in viral protein cleavage and genome replication. The wild-type virus also expressed NS5A protein, and the NS5A protein and double-stranded RNA co-localized in the cellular cytoplasm of wild-type transfected cells, suggesting active viral replication. Virus titer measurements indicated that the wild-type virus is infectious and produces over 10,000 foci-forming units per milliliter of infectious particle. At 6 hours post-transfection, both wild-type and pol-null viruses had similar luciferase activities, indicating a similar input level of transfected RNA had been translated. However, at 48 hours and 96 hours post-transfection, the wild-type virus exhibited increased genome replication levels compared to pol-null virus. The wild-type virus also produced viral NS3 protein. The pol-null virus had base-level luciferase activity, whereas the wild-type virus had a 2 to 3 log higher level of replication compared to pol-null reporter virus.
Once mastered, hepatitis C virological assays can be done in 2 weeks if they are performed properly.
While attempting this procedure, it's important to have robust cells and high-quality HCV genomic RNA transcripts. Following this procedure, characterized HCV strains can be tested in animal model systems to investigate in vivo fitness and host pathogen interactions. The development of an infectious HCV cell culture system paved the way for researchers to explore the virion morphogenesis and viral egress steps of the HCV replication cycle.
After watching this video, you should have a good understanding of how to perform various virological assays for characterizing different stages of the hepatitis C virus replication cycle. Don't forget that working with hepatitis C virus can be extremely hazardous, and biosafety precautions such as wearing appropriate personal protective equipment should always be taken while performing this procedure.